I was told that tonight is going to be a presentation by two extraordinary people for the things they have done. A faculty member and somebody he mentored. Is that roughly right? No?
She went to...
I'm a student. I graduated here in 2003.
Wonderful.
And then got my...
Okay.
My student.
So you're the mentor, okay. What I will tell you is, you're going to have a hard time persuading me you are more remarkable than the people I just saw. So there's a challenge to you. Now, they were very kind to me. They realize I'm from political science, so I have a very small brain. But what's amazing to me about this, and this day, is not just the degree of science and how small the world is that these scientists, in which these scientists work, but the enthusiasm that they can, with which they can describe what they do, and the potential for what they see. That to me, it's hard to put into a political scientist's brain, because it's so small, but at the same time with such incredible consequences. That if something holds, if this can be done, if we find this is true, if we're able to do that at this such a small level, then these things are possible. So in some degrees it's theoretical, but it's remarkably, amazingly precise and practical. And so to me today, it's been an eye-opening experience for how remarkable our faculty are, how remarkable our students are, the commitment they have to perseverance and success and not giving up, despite the hundreds of trials and months of waiting for the research. But something is pushing them to figure it out. Not everyone's going to go on and find the answer, or the new compound, or the new solution to something. But it's going to be because we have students and faculty and graduates who have that willingness to keep going at the very small level to produce something so magnificent. So I'm really eager to hear the presentation tonight. I'm thankful to be here. I can't wait to get started. Thank you.
As a provost of the university, there are certain things that make a provost very happy. The first one is when we see the students coming through the portal to the university, because it tells us that they are coming for their world to be transformed, their horizon to be extended. They are coming for their minds to be made open and richer. And then the second thing that happens that makes the provost happy is when the faculty follows them and drives in to the campus. Because otherwise we would just have students without the faculty. And then the third thing that makes a provost happy is when a provost looks at the faculty closely working together with the students in the classroom. Because you just know that that relationship produces something spectacular. It's like that chain of generations, okay, bounding together for the purpose of transforming society. So if you look at the civilization that we have today, that civilization is born out of that chain of faculty and students working quietly, extending the boundaries of known and unknown. It is remarkable that the achievement we have today in this world is born out of that relationship. So the most precious thing that to me is to see that master scholar and that the students working together, knowing too well that this faculty is handing over the mantle to that neophyte. And then the neophyte takes microphone and then they begin to speak like scholars. And then at that time, the faculty just sit down there smiling. It is the only trade, the only industry that I know that a faculty presents something through their research, something spectacular, and then they invite the students to come and critique it, and then the students come and critique it, and then the faculty is happy that their theory has been thrown out by their own students. And the reason is because it is an intellectual place where somebody once said to me, respect them who are searching for truth, but flee away from those who claim to have found it. So it is that joy that brings us together as intellectuals, a place where we are forever extending the boundaries of unknown and the known, searching for truth. It is that joy that when I look at the faculty and the work that they do. I came from a research university. It was a very cold environment where students have to fend for themselves and do things. When I see the faculty working closely together with the students, one-on-one at Arcadia, the first thing that came to my mind when I came here is, Mr. President, this is a very expensive thing to do, because at Kent State, you just teach to hundreds of students and walk away. But here to do it one-on-one with students, it is very expensive, but it's the most rewarding experience ever. So it is a joy for me, really, to introduce John Hoffman, that who I have known closely as an administrator, working very closely with me. And also, I see his passion for his students and working very closely with his students. It is that product that you're going to see tonight. When you hear your students, when you see the student talking and making all those presentations, know that what has happened is their interaction, okay, faculty-student interaction resulting into that. And just picture in your mind one day in Washington, D.C. or New York, big audience, and these scientists are speaking, addressing the whole world, talking about great things that have happened in their lives, starting from Arcadia. So please put your hands together for me to welcome John Hoffman to the podium.
I'd like to welcome you today to today's Faculty Student Research Expo. We're extremely happy that you're able to celebrate this event with us today. As our president indicated, we have a lot of opportunities this afternoon to go and visit our students and our faculty members in their own laboratories. So down in Boyer Hall, down in the Health Science Center, you have the opportunity to see where it is that the students are doing the work. And it's exciting to actually see where, it's exciting to see it here in a presentation venue, but it's even more exciting to see the students, to see them interacting with the pipettes and the microscopes and maybe the spiders, but we'll keep those in another place. But to see the passion that comes out because of the opportunities that they've received. I'd like to emphasize that what we're celebrating here today is some of the accomplishments done by our faculty and our students in the area of sciences. But it doesn't end here today. We've got students in the sciences doing work throughout the year. We've got a number of students that are going to spend their summers here, in many cases volunteering their time to be able to have the opportunity to get greater training in these areas. And that's an important amount of dedication for our students to be able to do that. Many of the students that hopefully you saw this afternoon are going to be taking the work that you saw presented at a preliminary point as their senior thesis projects or capstone projects. Many of these students are going to have the opportunity to present either locally at scientific meetings like the Sigma Xi conference, but many of our students have the opportunity to travel with their faculty mentors and go to national and international scientific meetings and to be able to present their work, the work that they're doing here as undergraduate students at Arcadia University, to scientists and scholars and researchers from around the world. And as Provost Michael indicated, to be critiqued, not by a peer, not by another student, not by a faculty member, but I've had students at a meeting and introduced them to Nobel laureates. Not because I knew them, but I saw the name tag. But that is a unique opportunity, and that's something that's incredibly important. and it's something that's incredibly valuable that we provide for our students. Again, we're emphasizing the sciences here today, but I would like to really point out that the types of hands-on experience, the scholarly activities that we're talking about, don't begin and end in the sciences. Even this evening, and I wrote it down, I lost my notes, the theater program has got a production of Girl Science going on later on this evening, from 8 to 10 in the Spruance Fine Arts Center. Just last week, the student biennial art exhibit closed. It had been open for 2 weeks. It's a juried exhibit. They bring in an outside expert to interact with the students. So the types of things that we're emphasizing today in the sciences are occurring in all of the different disciplines here at Arcadia. And again, I'm not going to do too much of a commercial, probably done it too much at this point, but if you get the opportunity, I encourage you to come back. Our capstone events start the end of April, they go into, I think, the second week of May. But this is an opportunity to, again, interact with students and see the projects that they've done across all of the different disciplines. And what's really spectacular is that I'm a biologist, I'm trained as a scientist. I know what science looks like, but to talk to an art student, to talk to a theater student, to talk to an English student about what they're doing within their discipline, and talking to them about the fact that their faculty member in whatever field that they're working in took the time to interact with them, took the time to get to know them, to provide them with this opportunity. And as Provost Michael said, it's an expensive way of doing things, but it makes a big difference in the lives of our students. And so I hope that you see this as a valuable opportunity here at Arcadia. It's not something that you find at a lot of other institutions, and it's something that we're very proud of. Now, the preceding commercial was brought to you by the Provost's office. It gives me great pleasure to introduce our first speaker this evening. Dr. Andrea Ketscheck is a stellar example of why research opportunities are so important for our undergraduate and graduate students. Many institutions, these opportunities are only valuable to the graduate students. We've got undergraduate students doing spectacular things on the lab. During Andrea's time as an undergraduate student, she spent several times in the laboratory of an evil scientist who's here, both on campus as well as completing an internship at the University of Pennsylvania's School of Veterinary Medicine. And when she graduated from Arcadia with a bachelor's degree in 2003, she already had 2 peer-reviewed scientific journal articles that she co-authored, representing the amount of work and dedication that she put in. She was in the laboratory working on a daily basis, and she was dedicated. She was there week in and week out, all the way through. And these were publications that she earned based on the work that she put in. After leaving Arcadia, she completed her doctoral degree at Drexel University College of Medicine, and she's currently a postdoctoral research fellow there. And what's really exciting for me, I guess kind of ages me, she was my student many, many years ago. Not that many, she's not that old, but she came back this semester and is an adjunct professor in the biology department, teaching in our interdisciplinary science program. So she started out as a student, became a student researcher, student scholar, got her doctorate degree, and now we got our claws in her, bringing her back, and she's teaching the next generation of undergraduate students. Please join me in welcoming Dr. Andrea Ketscheck.
Thank you, John, for that introduction. I also want to thank the committee for inviting me tonight and allowing me to speak to you about my research. Before I actually start on my presentation, I really enjoyed today looking at the students and their research and how they presented it with so much enthusiasm. Back when I went to school here, it was about 9 years ago, there was very little opportunity for research. And I never thought I wanted to be a scientist. I had dreams of being a medical doctor, a veterinarian, or even a forensic scientist. However, what I look back now is what intrigued me about those professions was there was always a question to be answered, always a problem to be solved. And I found myself always in the lab, as Dr. Hoffman said. I was in there on the weekends and during the day, because to me, it wasn't a job. It was something I enjoyed doing. And when you really enjoy something, you need to hang on to that. So that being said, I'm going to show you a little glimpse into my life as a scientist at Drexel College of Medicine. I graduated here in 2003, went on to get my PhD at Drexel, graduated from Drexel in 2009. How do you turn this on?
Little window.
So wait one here. Sorry. I should have woke it up a while ago. Anyway, so I'll just talk in the meantime until that comes up. But there is a lot of research going on in Arcadia right now, and I'm very impressed to see how many labs are conducting research and how many students are in those labs. I think when I graduated, there was probably only like a handful, maybe 1, 2, or 3 students going on to graduate school. And every student I talked to today had the ambition to go on and continue their education on graduate school. So, the title of my talk is the cytoskeletal and signaling mechanisms responsible for axon collateral branching of sensory neurons. The nervous system is responsible for everything you do every minute of the day. It is responsible for doing simple tasks such as walking your dog or reading a book. And then it is responsible for more complicated tasks such as figure skating or theorizing in physics. The nervous system is composed of 2 main compartments: the central nervous system, which controls your brain and your spinal cord, or your peripheral nervous system, which is the nerve endings that reach out to the body and communicate between the brain and the rest of your body. It is the neurons that are the specialized cells involved in the communication between your brain and the rest of your body. This is a schematic of the structure of a neuron. So you have, each neuron has a cell body, which is the soma, which is the metabolic center of that neuron. The neuron then will receive information through the dendrites and then send information out to the axon. This information will be sent to other types of cells, such as other neurons to communicate with each other, or cells of your skin or your muscles so that you can feel or move or walk. Now, the neuron has a basic cytoskeleton, which is common to all types of eukaryotic cells. It contains microtubules, which are predominantly in the axon shaft, and control the transportation of molecules to and from the cell body. It also contains actin. Actin is going to be the majority of my talk today. So actin is involved in the morphological structures of the neuron and the motility of the neuron. It's going to be very dynamic. Now, axon collateral branching is something that interests me and my lab right now. So what is axon collateral branching? Axon collateral branching is when a neuron contacts multiple cells from one neuron. So if you think about it, there's probably about 150 billion cells in your brain. However, those cells need to make contact to over 100 trillion cells in your body. In order for this to be accomplished, one neuron must send out multiple branches to different cell types. Now, collateral branching is not only important during development, but also when the nervous system suffers an injury or a disorder. And for example, collateral branching will be important in spinal cord injuries, brain injuries, multiple sclerosis, strokes, and Alzheimer's. Now, how this happens is, for instance, you have cells talking to each other right here and communicating. This cell in blue may suffer an injury or a cut, where the axon is then drawn back from the target cell. Cells close by in the vicinity of this injury can then sprout off axons off of the original axon, producing collateral branches to regain function of that nervous system. So our lab is interested in studying sensory axons. Sensory axons are a dorsal root ganglion. They are located right off of the spinal cord, And what's important about the sensory axons is that they will send an axon down and into the spinal cord and also out and into the periphery to attach to your skin cells. These sensory neurons will then send collateral branches. So if you notice this neuron in blue, it will send an axon into the spinal cord and then break off into different collateral branches. In our lab, we use embryonic chick day 7 to study collateral branch formation. We dissect the DRGs out of the chick, and then we culture them either as a DRG explant, right here, or as dissociated cell single cells. We also transfect them. Sorry, that video is not shown, but I'll show you that video again. We will transfect the cell so that we're basically infecting it with DNA that's conjugated to a fluorescent so that we can actually visualize that protein in the cell and watch the dynamics of that cell. So collateral branch forms from phylopodia. And what are phylopodia? They're these little tiny structures coming off of the axon. They're very dynamic, and in some cases this little tiny structure will elongate. So this is a time-lapse image, and you can see over time this structure right here elongates, and eventually forms a growth cone at the tip of the structure, producing a new collateral branch. Nerve growth factor is a small secreted protein that is critical for the survival, differentiation, growth, and maintenance of sensory neurons. It is released at these target cells and is responsible for promoting collateral axon branches. The contact with the nerve growth factor induces the phylopodia formation. What my goal is to understand is what is the cellular mechanism by which NGF induces axonal phylopodia. So our lab has recently described a model in which how phylopodia are formed. And here you can see it's a 3-step model. So this is an axon pseudo-coloring image in which the light blue is going to be a lower intensity of actin, and the red is going to be a higher intensity of actin. And you can see that over time, actin will accumulate into these patches and eventually produce a phylopodia. Now what's important is that all phylopodia come from a patch. However, not all patches produce a phylopodia. Only 20% of patches will produce a phylopodia. This is extremely important right now because if we can figure out what that key transition protein is, or that signaling pathway, we will then be able to develop certain drugs that will promote collateral branching when the nervous system suffers from injuries, such as spinal cord injury. Now we wanted to know, since NGF, we see here NGF increases the number of phylopodia along the axon. So if you take a look at this figure over here, this is an axon with the growth cone of the neuron. And you can see without NGF, there's few little actin patches and very little phylopodia. You treat the neuron with NGF, the nerve growth factor, and you will see an abundant amount of actin patches and phylopodia extending off of the axon. I wanted to know what stage is NGF affecting? Is it affecting that initiation stage? Is it affecting the elaboration stage? Or is it affecting the transition stage? Now, so here's that video I was going to show you earlier that didn't quite work. But this is live imaging of the neurons in culture fish. And these are transfected with, actin. And so you can see that the actin patches will develop and then produce phylopodia along the axon. So after looking at a couple of these videos, you can then analyze. And what we found was that NGF increases the number of patch formation. It has no effect on that transition or the elaboration. This is another video with pseudocoloring, and you can see that in blue, you have lower intensity of actin, red, higher intensity of actin, and the high intensity of actin will give rise to a phylopodia. So now that we know that NGF causes an increase in F-actin patches, we wanted to know the signaling mechanism. Some people get a little overwhelmed when they see a diagram like this. Believe me, I do too, and I have my PhD in cell biology. So that is why I kind of tried to highlight this little area right here. And what's best about cell biologists is, yes, there is a lot of signaling pathways. But what you need to do is kind of just pick it apart. And you need to just say, I'm going to concentrate on one section first. You can't just solve everything in one day. So first off, we decided to look at PI3 kinase and AKT, this little tiny pathway right here. And so we know that NGF activates TrkA receptors, which then will activate PI3 kinase. PI3 kinase phosphorylates PIP2, PIP2 increases PIP3, which then binds the pH domain. That sounds a little confusing. When I told my dad that, he was like, you lost me. So what you need to know, though, is that it is the pH-AKT domain right here is going to be conjugated to a fluorescence. So you're going to be able to visualize it in the neuron. And what I want to see is if that domain is the same area at which the actin patch is localized to. So we transfected the neurons with this pH-AKT domain. And you can see that they are actually localized right at the tip of phylopodia. So we wanted to see, oh, okay, do they co-localize, meaning do they exist in the same spot as actin patches? And so we co-transfected with both pH-AKT and actin, and we found that yes, they do coexist and co-localize in the same exact spot. Not only that, they rose at the same time and they disappeared at the same time. So these two molecules are very closely related. So that suggests now that NGF is acting through the PI3 kinase AKT pathway, but it doesn't really solve that solution. In order to really stress that, we need to inhibit that pathway. And so we inhibit using pharmacological drugs, we inhibit PI3 kinase and AKT. And what we found was that when you inhibit PI3 kinase, you block that NGF increase in F-actin patches and phylopodia formation. Also, when you inhibit with AKT, you block that NGF increase in F-actin patch and phylopodia formation. And lastly, the second thing you can also do, so not only have we localized the pathway to the actin patches, we've inhibited and blocked the pathway, but the last thing you can do is now activate the pathway. And so we decided to activate the pathway using a peptide that was synthesized for us to activate the PI3 kinase pathway. And in this image here, you can see this is labeled for tubulin, and you can see the axon has very little collateral branches coming off of it. However, with the PI3 kinase peptide, there are a lot more collateral branches extending off the axon, and they are longer. Now, more importantly, when you analyze this, what we found was when you treat with PI3 kinase peptide and NGF, you get no additive effect. It is the same effect as if you were treating with PI3 kinase peptide or treating with just NGF. What does this mean? It means that they must be acting through the same signaling pathway. Because if they were both acting to increase F-actin patches but doing different separate pathways, then you would see an increase with the PI3 kinase peptide and the NGF. So in summary, in case I lost any of you, NGF increases the formation of axonal phylopodia by increasing the frequency of F-actin patch formation. NGF acts through the PI3 kinase AKT pathway. Now, I just showed you a little tiny picture in here in red. But really, what we have now going on to do is showing all this. Of course, that would probably take me close to 3 hours to tell you everything I've done in the past 2 years. But what I need you to know is that the signaling pathway has then gone and led on to different molecules involved in the actin patch formation, also looking at cytoskeleton, such as microtubules, and more exciting news is what we're looking at right now, and is the first to look at it, is protein synthesis, local protein synthesis in the axon. As I told you before, the axons are sometimes positioned very far distances from the cell body. And you can imagine if that axon is receiving information, it's going to take a very long time for that information to get back to the cell body, process it, and then go back to the growth cone and start changing its behavior. We are now noticing and doing some experiments showing that there is actual local protein synthesis and ribosomes in the axon, which has never been shown before. So here's my quick little picture that I just got last week, and this is the little red dots are showing that ribosomal RNA are at the base of actin phylopodia. So, and last thing I really just want to show you, because it's very exciting for us in the lab, is a lot of the questions they ask us is, so you see it in a dish. I'm a human. Well, how do you know that's going on in my body? So we developed this technique called in ovo electroporation. And like I said, we use the chicks to study collateral branch formation. And so instead of taking the chick out and taking the neurons out, what we decided to do is to transfect those neurons in the chick. So here you can see the chick in the egg. You just open the egg, you see the chick, you're going to inject your plasmid or your DNA right into that neural tube. You're going to close that chick back up with tape. Just cover the shell with tape, which is amazing. I thought that it would die with bacteria or something. But no, they end up surviving, they develop perfectly normal, and then 3 days later you can take it out and you can actually see your neurons growing fluorescently green through the limb of the chick. Another thing you can do is just take out the spinal cord and look at your axons right in the spinal cord. Now, we wanted to see, of course, all of our research is based on that patch model, and we wanted to see if that existed in ovo. And we are able to see that this is a time lapse, and you can see the pseudocoloring, which is the light blue, which is the low intensity actin, versus red, the high intensity actin. And you can see a patch formation forming over time and giving rise to a phylopodia. And this is just another picture showing how clear you'd be able to see the phylopodia using phase or something in ovo. So what does this mean? This means that this is important research because understanding signaling pathways, I know it looks very confusing at times, and I don't even know how I do it. I don't think I'm that smart, but somehow my brain just clicks this way, and I can look at something and want to figure it out. It looks confusing, but understanding the signaling pathways is extremely important because that's the way we're going to be able to develop new drugs that can then be applied to nervous system disorders or injuries, and we can either block that signaling pathway or activate it, hoping to produce more collateral branches and recover function once it's lost. So that being said, there's a couple of quotes I really enjoyed, and we can't solve problems by using the same kind of thinking we used when we created them, by Albert Einstein. This is the lab of which most of all this work is done, and of course I would not be here today at all if it wasn't for my mentors. Actually, to tell you the truth, I would not be here if it was not for Arcadia. I never wanted to be a scientist. I never even thought of being a scientist. Until one day, Dr. Ramachka said, Andrea, I think you need to meet someone. And his name's Dr. Hoffman. I think you'd work good with him. And I was like, research? Rats? Are you kidding me? It ended up being the love of my life. And I think it's great to be able to wake up every single day and love going into work and love doing what you love doing. It's not a job to me. And as Dr. Hoffman said, I was in the lab all the time, but that's because I enjoyed being in the lab. And I think it's great that Arcadia is having more opportunities for students to do research, because research is a big part of science, and it should not be overlooked. And that's...
I'd like to thank everyone for coming out tonight. Before I talk about what our Arcadia undergraduates are doing with invasive species here at Arcadia, I want to speak briefly about everyone's favorite species, that being humans. There's currently quite a lot of us on the planet, about 7 billion. That's really a huge number. It would actually take you 222 years to count to 7 billion if you did one digit up per second. And that's projected to grow to about 10 billion by the end of the century. And one thing all these people like to do is travel, go see the world. That is definitely part of the Arcadia undergraduate experience. We also like to trade a lot of merchandise with other countries. So there's been no other time in our history where the world has been so connected by the movement of people and the movement of merchandise. That's generally a very positive thing. However, it has had some negative consequences, such as globalization has allowed many species to be introduced to new environments. This can be intentional or accidental. And when this happens, let's say an insect comes over from Australia to Pennsylvania, most of the time this new species is going to die, right? It's just not going to make it. Philly's a tough town. Right? Some will establish, and a small percentage of those become what is known as invasive species. And they have a pretty simple definition. These are just organisms that are non-native to an ecosystem whose introduction causes economic or environmental harm or harm to human health. There's quite a lot of examples of invasive species. I'm going to talk just about a few of those here. The grass carp, native to China, has been introduced throughout the planet to control aquatic vegetation in lakes and rivers. It does a great job. It eats everything it can get its mouth on. And one of the bad aspects about that is that they actually transmit tapeworms to our native fish and have been reducing their populations. And once they eat all the aquatic vegetation, they tend to facilitate the rise of harmful bacteria like blue-green algae, which can be toxic to human beings. And they can reach fairly high population sizes. A more recent example is the Burmese python, which has become quite invasive to the Florida Everglades. And these are actually the descendants of pets that were released into the Everglades, and now their populations have just exploded. And there's a recent paper suggesting that every medium-sized mammal in the Everglades is on the verge of going extinct. So how does this happen? How do these populations just start to skyrocket? Well, there's a few ideas. One being the escape from enemy hypothesis, and that suggests that when a new species comes to an area, it leaves its predators and parasites behind. So it's basically in a predator-free zone, and its populations just, start to explode. There's also the ecological meltdown hypothesis. This is where one invasive species facilitates the spread of another invasive species. And the classic example is where you have, say, invasive sunfish, which are going to decimate local populations of native aquatic invertebrates that, say, live in a lake. And these native invertebrates tend to keep the populations of invasive bullfrogs in check by eating their tadpoles. So if you have more invasive sunfish, you have less native invertebrates, more invasive bullfrogs. So the sunfish is actually facilitating the spread of the bullfrog. There's quite a lot of invasive species in the United States at this time. They definitely are outcompeting and consuming our native biodiversity. There's some pretty severe economic losses to our economy and to also the global economy. You have one more example. Here's some forests in New Jersey that are being decimated by the invasive gypsy moth that was introduced from Europe. Now this is going to be the focal species for the talk, the Asian tiger mosquito, or Aedes albopictus. Right? It's native to southeastern Asia, but it's invaded 28 countries since the early 1980s. Its predicted future range is every tropical and temperate country on the planet. And the way that it spread is through the international trade of used tires. Apparently there's a big trade in used tires, right? Shown here. And it's not that these females, which are pretty striking animals, right? They definitely are transmitting quite a lot of viruses to humans. It's not that you get females and males living in the tires. The tires transport eggs. Here's the eggs shown right here, and I had these out in the lab today. They're desiccation tolerant. They can withstand drying for up to 1 year. So you get these eggs and these tires that are just spread around the planet. They also love to breed in artificial containers, like say a beer bottle that somebody throws out into the woods, or not only an old tire or just a bucket that fills with water. It only takes the larva, shown here, about 5 to 10 days and 1 centimeter of water to become an adult. So because of this, they tend to be super abundant in urban and suburban areas. Here's an example of some of their breeding sites. It just doesn't take a lot of water to get this species to complete its life cycle. So you just have to wonder, how many breeding sites could you have in an area like this? Right here's Philadelphia. Here we are right here. I don't know the answer to this question. More than 10. Right? So, quite a lot. But luckily, humans being the sophisticated species that we are, we've developed some pretty ingenious ways to fight the Asian tiger mosquito. That being with the use of biopesticides. These are just naturally occurring bacteria, such as Bacillus thuringiensis israelensis. This kind of rolls off the tongue right there. I'm just going to call that BTI for this talk. And also Bacillus asparagus. I'm just going to call that BS for the rest of the week. Right? And what's cool about these bacteria, they produce highly toxic proteins that are very specific to mosquito and black fly larvae. So here's a BTI individual right here, this naturally occurring bacteria. So what happens is mosquito larvae, which eat bacteria, when they consume these individuals, these proteins, shown here surrounding the spore, actually embed into the cell membranes of their intestinal cells, generate these pores. All this water rushes into the intestinal cells. Boom, they actually burst, and they're going to die soon after that. And it's actually just, it's very common. You can buy this anywhere because it's very safe, very low toxicity to anything besides some mosquitoes and some other dipterans. And it dominates global efforts to control mosquitoes. This is how it's done around the planet. It's the most dominant way we're attempting to keep mosquito populations in check. So to give some brief natural history of the mosquito, right, here's a female. She's taking a blood meal. Right? She needs a blood meal to complete egg development, and this is where the viruses get transferred to humans. For Asian tigers, the eggs are actually laid not in the water. They lay right above the water line, but they can just lay in a damp area. When it rains, it floods. The eggs hatch out. The mosquito larvae shown here. If all goes well, there's a pupation stage, and back to an adult. And most of the research with mosquitoes and how to control them is emphasized this stage right here. How do we kill the larva? But what the undergraduates have done is looked at where the females are laying their eggs, right? So it's a slightly different twist to an old problem. It's a subtle and important difference. So not only are the undergraduates quantifying how many eggs the Asian tiger females are laying, again, here's the egg shown here, and only about 1 millimeter long. They're also going to be quantifying egg rafts. Here's a native white spotted mosquito laying an egg raft on the water. Any one egg raft has about 200 to 300 eggs. It's called the white spotted mosquito because it has these white spots. It's a fairly non-aggressive species. We most likely have this in your basement, your attic, or your shed right now. They don't feed on people. They prefer actually to feed on birds. So our undergrads have been able to quantify this species as well. They also like to breed in very small bodies of water. So what have they been doing? Well, there's a whole group of undergraduates, including Sneha Thomas, shown here, and she asked, how do invasive Asian tiger and native white spotted female mosquitoes, how do they respond to biopesticides at breeding sites when laying their eggs? So we're going to put out these breeding sites with biopesticides. We're going to see what the females do. Do they select them? Do they avoid them? Or what have you? Then she's going to look at how is larval survivorship affected by these same biopesticides. And our methods are pretty simple. We actually generate these experimental breeding sites, which are just these black Dixie cups, shown here, filled with some water. There's a tongue depressor wrapped in a brown paper towel. And then this is where the female is going to lay their eggs, right? And we can go out, we can collect the sticks, count the eggs, count the egg rafts, and do some pretty standard statistical analysis. But during the summer, you're going to see lots of these on campus. It's all laid out. We just manipulate what happens inside the cup and see, well, how are the females going to respond? Here's our study site. This is Arcadia. We mainly work down by the creek, right along the creek. So what did Sneha get? Here's the mean number of Asian tiger eggs on the y-axis for these different biopesticide treatments. We had none, our control, so there's no biopesticides in the cup. We had 0.1 grams of BTI placed in there, or 0.1 grams of BS placed in. We did a combo of about 0.05 grams and 0.05 grams of both pesticides. And what she's shown is that Asian tiger females actually avoid, there's a significant reduction in the number of eggs laid in any cup containing biopesticides. Most of the eggs are laid in the controls. When you look at the same thing in the culex, the native species, here's the number of egg rafts on the y-axis, the same biopesticide treatments. The native white spotted females actually select. They lay more egg rafts, significantly more egg rafts, in any of our breeding sites containing biopesticide compared to the controls. And this is two different experiments, so it's not like they're actually being affected by each other. Then she looked at larval performance to see what happens. You know, what's the consequence of laying your eggs in these sites? Well, it kills them. Here's it does. This stuff works great. So for the Asian tiger females, here's the percent larval survivorship on the y-axis, same biopesticide treatments. They all die except in the controls. Same finding for the culex, right? Their larval survivorship here. It kills them all, right? This is actually really good stuff. We have a whole other set of undergraduates been looking at some other questions, like Ashley Zeckman, shown here. I think Ashley is in the audience right there. Right? And what Ashley asked for her senior thesis, how do invasive Asian tiger and native white spotted mosquitoes respond to both invasive and native plants in breeding sites when laying their eggs? She's basically testing the ecological meltdown hypothesis. Here's one of our breeding sites shown right here. This contains actually an invasive grass, Japanese stilt grass. So the idea is, well, you have these Asian mosquitoes, we'll get some Asian invasive plants, and maybe actually they will, facilitate. Maybe having the grass in the water will cause more female Asian tigers to lay their eggs. But that didn't happen. Actually, we got the complete opposite result. Right? Here's a number of Asian tiger eggs on the y-axis. We had different plant treatments, none, no plants. We had native grass, cut grass. We had an invasive Japanese stilt grass. We had an invasive grass actually from Europe, reed canary grass. The Asian tiger females are just not laying significantly less eggs in any of our grass treatments. It doesn't matter if they're native or invasive, they're just avoiding these grasses. She did another experiment with our native females, right, from the white spotted species. It kills egg grafts on the y-axis. These are plant species. They actually lay significantly more eggs, more egg grafts in any of the treatments containing any grass. It doesn't matter if they're native or invasive. We're just getting different responses. Right. There's another student named Will Rocco. He did a similar study looking at a little more complex. We put out his breeding sites in the shade, in the dark squares, in the sun, with the open circles. We had controlled no plant species, and he had a native polygonum species and an invasive Japanese knotweed was also in the same genus, polygonum. So we had a native and invasive plant in the same genus. They're very closely related. What Will showed is that they're going to lay more eggs in the shade compared to the sun, and they're going to lay more eggs when there's plants. This doesn't matter if it's a native plant or an invasive plant. They're actually just choosing to lay with this particular group of species. So invasive, native plant, it doesn't matter for their reproduction. Then finally, I'm going to talk about some work we do actually in class, either in a research methods class or aquatic biology class. We do some pretty complex experiments. This is actually the most complex experiment I've ever done in my life. And we actually asked, how do Asian tiger females respond to different light levels? We had shade and sun, different water levels. We had cups with lots of water, large and small amounts of water. And we crossed that with the presence and absence of a biopesticide. So that's 8 treatments. We replicated 10 times for a total of 80 breeding sites. It's just, something we do in lab. So it's not as if at Arcadia, we're going to do, simple experiments in the classroom. This is actually fairly complex. And they seem to actually enjoy it. We got some pretty complex results. Here's again, mean number of Asian tiger eggs on the y-axis. And I'm not going to go through all of the results, but what we can see that the females are doing, they will lay most of their eggs in the shade with small amounts of water. The cups only have about 200 milliliters of water. They really like that. And containing no biopesticide. That's what wins. Shade, small, no biopesticide. They avoid the strongest, the complete opposite mix of treatments. So the least number of eggs were laid in the sun with large amounts of water and contain the BTI and BS pesticide. So in summary, we had two egg-laying responses to the biopesticides. Asian tiger females avoid. The white spotted native females they actually select, but only one larval response, that being death. This is actually a very important finding. It suggests that there is a secondary pathway to resistance. It's not coming through larval survivorship. The females are being selected to avoid these biopesticides. This can greatly reduce their effectiveness. We had no evidence supporting ecological meltdown. And on some level, I guess this is good. It's not that we have all these invasive plants on campus that are causing the invasive mosquitoes to lay more eggs. With that being said, I was actually very much rooting for this experiment. You're not supposed to want your results to go anywhere, but the undergraduates are like, well, it's pretty cool. We have these invasive plants, invasive mosquitoes. Maybe they're facilitating egg laying, but it just didn't happen, right? So it's a good thing, but all at the same time, I was a little upset. And then no one really likes mosquitoes. I hear this a lot. And I don't have any great love for mosquitoes, but I do definitely respect their capacity as mothers to pick and choose amongst breeding sites, because this is their parental care. They're very good at where they lay their eggs, which is one reason they're so abundant. And a better understanding of where they like to lay their eggs can have increase our ability to control their populations. If we know where are the good breeding sites, those can be targeted. Like the end, just with the importance of this. Again, these are what people have been talking about all day long. Exposed numerous undergraduates to experimental biology. They're getting skills in experimental design, data analysis, and scientific writing. And some of the designs we do are fairly complex. And the math we use to analyze it can be used for any experiment. You can have an experiment with 2 drugs, look at drug interactions. You can use the same mathematics, right? So it's an important skills. But most importantly, right? Arcadia undergraduates do important research investigating real-world problems, and they do produce valuable results that benefit society. And I'm not just saying that because we have very important people in this room. It is true. It's true not just in biology, but just all around Arcadia. We have some really smart undergraduates here doing some really cool work. And there's always a lot of people you need to thank when you do any research. I'd like to thank you guys for showing up. And I don't know if we want to go to questions or we just want to go to dinner and have questions there. Either way, but thanks again.